All right, so you know the routine by now. If you have questions, just raise your hand, and I will be there with a microphone, and we'll make it work. So let me start maybe by asking you the following question. What do you think, what kind of technology or series of technologies is the most critical limiting factor today in understanding the function of genome in particular? What is, if you had a wish list, let's say we live in a simulation, and we actually have a chance to communicate with our masters, protein, yeah? So what should we ask them to do?
I have a short answer. Okay. And I'll preface this by saying that obviously we're trying to be Santa Claus rather than the people who actually make the scientific discoveries. I see this huge gap between the length and timescales that we can perceive and what's going on in a cell. There's 9 orders of magnitude difference in size, and things move a lot faster. And also the sorts of events that happen are outside of our ability to perceive. That's why imaging is so useful. But translating what's going on in a cell into interpretable images, I think remains, despite imaging is amazing now, but it's still a huge challenge. There's tons of development to be done, I think, and I'm biased.
So I guess my answer to that, and then this was part of a conversation from earlier, is that, the imaging, I love it. I don't want to have to do high-C. I'm very intimidated by it. However, high-C gives you information about what sequence, right? You can take the inferred spatial, closeness from the high-C assay and actually map it to genes, chromosomes, right? So that there's that connection that exists for high-C and not so much for the imaging, at least what you can do in a live cell, right? And then there come problems from fixation, right? So you could do FISH, but you got to fix the cells, right? And so so I'm not aware of any sort of like really high fidelity sequence locating technology that you can do in live cells. So yeah, that's up to you. That would be a really cool trick. And another thing I'm going to maybe mention, and then so this is getting into something that I've been thinking about a lot recently, which is kind of a callback to my PhD work, especially in a solid tumor. There are sort of real-time, very dramatic changes in the structure of the genome. Genomes tend to be very unstable in a proliferating solid tumor population. If you got anyone, in case you're not familiar, so there's a brilliant scientist, Dennis Disher, who's based at UPenn, and he looks at a lung cancer cell model. And so they've got a system to where they can actually detect entire losses of chromosome arms as cells are sort of like, contending with stiff ECM. And so it almost sort of creates like this evolutionary selection, right? Because there are some losses that actually appear to be beneficial to the cancer cells. So these things actually matter. And so now you've even got, we've got to develop tools that can contend with the dynamics of the integrity of the genome, right? You're probably getting changes in ploidy, right? You got to distinguish the different, sister chromosomes from each other, right? Looking at different alleles. And so I think that, being able to, actually link the physical data with sequence is the big problem to solve.
And I'll come at this with a little bit less experience, but my wish list is also related to sequence. So I work a lot with ChIP-seq data or high-C data, and I know that we're limited in the resolution of this data as well as how we handle repeat regions of the genome. So there's a lot of high-C contact maps I look at with big gaps, and I can predict structure in those regions using simulations, but I have no way to validate that structure. And that's been biasing my models. So a way to handle repeat regions would be helpful for me.
So following up on this technology theme, will we ever be able to identify, localize and label every single gene in the cell at a single cell level in 3D, let's say, and map it all out the way it really looks? Or will we always go back to sequencing techniques?
The question is whether we'll ever be able to do it?
Right.
Oh, yes.
And if you think we will get there at some point, I guess the question is, what is missing? What kind of critical knowledge or capability we still need to attain to get there?
I mean, I think it's a technology challenge, right? Because I mean, there are, right? So we know a lot about, right? So you can look to nature to look for proteins and things that seem to have, some level of sequence specificity. So I do know a few examples of, CRISPR, right, as an adapter, and some efforts in using that system to specifically label sequences. So actually, yes, so now I've just come up with a good answer, specific answer to that question. Even when you do that, if you're looking for like a promoter, right, something as short as a promoter, one guide RNA, one CRISPR molecule, you're gonna have a hard time unless someone can solve that problem. But then in the meantime, maybe a way to do a signal amplification, right? So if you manage to have some adapter or molecule interacting specifically with a sequence, and you can do this in a live cell, it's going to be really hard to see it, right? Because there's just so little to detect. So yeah, in terms of like looking at the more unique sequences, so repetitious sequences, I know there's been some really beautiful work sort of like painting middle repetitious sequences, also coupled with specific chromatin modifications, right? These sort of like, I think it's called BiAD or some sort of probe like that. But you can only do it with repetitious sequence to deal with that signal amplification problem. So to me, I think it would be really useful. It would really be a leap forward if we could use things like, I mean, even zinc fingers, tails, whatever you want to use, go after that site the same way you would send a regulator after a specific site. But instead of using that to regulate genes, you can look at it, right, and follow where it is in the nucleus. But that's beyond my expertise. Like, how do you deal with, having so little of that probe to look at?
Yeah, just to tag on to that, I totally agree with at least the parts of that I understood. And I also wanted to add that I think we know at this point that there are no imaging technologies that are completely non-perturbative. I certainly can't think of one. Even the smallest tags, vibrational tags, or native fluorescence, even just shining a laser at a cell, you're going to be changing its behavior in some way. But given that, and given that there are so many clever imaging strategies that have complementary information that you can glean from them, and especially when you pair that with sequencing and other readouts, I think that making all of these advanced technologies as accessible as possible so that they can be used to actually solve problems and learn things about biology and medicine, I think that's the real challenge, because I think that non-perturbative imaging of sequence-specific information might stay science fiction forever.
Well, and by the way, if you do have questions, don't be shy. Ask for the mic. You brought up a very interesting question of dissemination, right? Dissemination of technologies and whether there's a hardware or protocols or computational technologies. And I think I agree with you. A lot of things that we find really powerful and fascinating, we read about them in Nature and Science, but some of them, quite a few of them actually, stay in the lab that develop them. What is the reason for this, and how can we address that? What are the critical barriers?
I can approach this from a computational perspective. So we in our lab developed a simulator for our research. There are other labs like us who've developed their own simulators. but it's not too often that we actually use each other's simulators. And I think a big part of that is scientific communication and documenting our science so that we can generate reproducible results. So some steps towards that in the software space would be to publish not just walls of code, but maybe some notebooks that demonstrate implementation using the code. And that's something that we hope to do with the latest work.
Sure, yeah. I mean, you absolutely have to go do a good job documenting everything and getting it out there. But I think there are also natural stages of technology progression. When super resolution was first introduced in Xiaohui's lab and others, there were only those couple of labs doing it, but now tons of labs use it. It's much more accessible. So for our specific technology, for example, I know probably everybody who's built an Able Trap in the world. There aren't that many of them. But there are a lot more now, and we have people asking us how to do it. So, I think there's just sort of a natural life cycle to getting these technologies accessible for everyone to use.
I think I'll add, even I guess what would be relatively less fancy assays, Hi-C, ChIP-seq, we still struggle with. So we're interested in sort of like breaking chromosomes down to just the right level of subsegments. Well, how do you define that? So my grad student actually spent quite a few days looking at how, different, like from paper to paper, so apparently for these TADs, right, there's like an insulation score, and the insulators are there depending on what threshold cutoff you use. And it took us a while to actually come to that realization, because that nuance wasn't really discussed or disclosed. So even with some assays and technologies that have been around for a little while, this practice of, having consensus around, best practices for interpreting the data are still needed. And I know people are discussing these things, but that work is very important. So I just want to, say that to encourage people to continue thinking about that and working on that.
Hi, thank you so much, you all, for such an interesting afternoon. I had a specific question for Joe. May I understand a little bit better about the crosstalk interaction? What is biologically the significance of that? It will help, it's like amplifying the shutdown or the increasing the capability of expression of the nucleosomes. What is the biological effect of that? And then I think the other question is, how would you predict that to be if a cell is more plastic? So if it is capable of, upregulating, downregulating, what would you expect to happen under that circumstance? Thank you so much.
That's a great question, and that's kind of related to vignette number 3 that I didn't get to during the talk, and that's about how stable different epigenetic domains are. So we developed a model to study how marks spread over multiple cell generations related to a phenomenon like epigenetic drift, which has been linked to age-related diseases. So over time, our epigenetic marks are changing, and that's destabilizing the chromatin fiber. So if there's a crosstalk, a synergistic crosstalk between different epigenetic marks, perhaps these marks can stabilize one another in a way that increase the fidelity by which epigenetic profiles are maintained, and that's an area of research we're interested in going into in the future. Quite honestly, I'm not sure how this would play into the plasticity of a cell, or how cell plasticity affects that.
So then, if you don't mind, maybe I missed this point. So crosstalk, you think you would tend to shut down more the transcription, or could go either way?
In our models, we're looking at repressive reader proteins, and for these, it'll shut down.
Shut down. But you would expect the same thing could happen.
So
you could activate faster So through crosstalk, even if the particular histone is like not the perfect match, you could say that this could be like an amplifier of shutting down transcription or upregulating transcription. It could both methods could happen, you think. Would you predict that?
So I think this depends on the mechanism by which these different reader proteins are affecting chromatin structure and facilitating gene regulations. I think for repressive reader proteins that have this binding and oligomerization mechanism, there is evidence of a crosstalk that represses the gene expression. I'm not familiar with a similar mechanism for gene activation, but I would, if I were to take a guess, I would imagine there may be some out there where one type of either methyltransferase or another protein that affects the epigenetic landscape is recruited by a bound reader protein, and this would be some sort of crosstalk that could spread activating marks.
Can I add, I guess maybe put on the hat of the audience with that? So that I really like that question a lot, because to me, that has me thinking about this concept of bivalency. And then so H3K27 trimethyl has been associated with these regions that are known to be bivalent. Part of that definition of bivalency is that, okay, so you're kind of sort of maybe express a little bit, and then if you do something in that bivalent region, right, it's K27 trimethyl and K4 trimethyl sort of coexisting in the same space. One of the observed phenomena is that if you stimulate repression, you kind of flip this switch and you toggle over to stable repression. And then so what I was kind of excited to see was your, evidence or suggesting that there's crosstalk between polycomb, which kind of is like at these poised genes, but then you're potentially bringing in HP1, which is associated with a more stable repressed state. So that could help to maybe explain how bistability switches over, commits to one side. Yeah, so we can talk later.
Actually, maybe we should have
sure.
conversation offline. Yeah. It would be interesting to talk more about that.
Yeah.
Okay, we have a question right here.
Sorry, I didn't mean to grab the mic.
No, you're fine. Yeah, I was going to
So in today's talks, one of the challenges still remains to be going from the imaging to the genomic sequence. So I'm not an imaging or physicist, but I'm a genomics person. It looks like one of the challenges there was the number of colors that we could have. But it looks like you have solved that issue at this. Like with the genomic sequence, if you have 20 nucleotides, then we can align it to the genome. So you seem to have 27 colors. So I guess that challenge at least is not there. So the next challenge is how are we going to, so what is the physical kind of space that we can bring all these different colors next to each other? If, I assume that, they cannot work next to each other, but is there a way to kind of make them photo-activatable or photo-switchable? Then we can sequentially kind of activate them, and maybe then we can align those colors to the genomic position, potentially. I don't know if it makes sense.
No, thank you so much for the question/suggestion. It feels like you're writing my grants for me. Yes, you're right that, okay, fine, we've got a bunch of colors, and we think we can, Kavya's, is your poster showing the, 50-something? Okay, well, we can do a lot more than what we're showing in this paper, but all of that is in this confocal flow cell geometry. It's not imaging. But what we're trying to do next is the imaging and figuring out, of course, the diffraction limit tells us how close together things can be, but there's a separate question of, we're not going to have the same precision that the Abel trap has on telling things apart, but how many can we tell apart? And if we add, there are great techniques to do wide field FLIM, even for single molecules now, lifetime imaging. And if you add that, can you do a little bit better? And so what we're envisioning is being able to combine these fret floors that we've designed with techniques like DNA paint or other super resolution techniques to make those at least an order of magnitude faster or better or more multiplex, depending on which axis you're trying to affect. It gets complicated very quickly. And I think any of those things, okay, you can't track every gene all at once, but if you can track 10 at once, that's pretty good. And then, if now you don't need 10 rounds, you need right, or you don't need any rounds. And if you can photoactivate so that you can, maybe have some timing, it gets complicated really fast, but that's part of why we're excited that we were able to create different colors with something kind of simple. All this to say, yes, and this is an area that we're actively exploring. Thanks.
Okay. Thank you for the talks, and I have a question for Alison. That's your talk. I think I understood a little bit. When you were presenting, multimodal fusion or multi-view learning came to mind from machine learning. Or alternatively, in machine learning, you have the, kernelization trick where you take something from a low-dimensional space and you map it to a high-dimensional space where separation can be linear and all that good stuff. So, that's great. What I missed, you keep saying tracking. So, can you really tell where each and every molecule or particle is over time? It seems like with trapping, you lose that information, right? So that's my question. So can you, again, show the tracks over milliseconds or whatever the time interval is of 10 particles? That's what I missed, but the question is if you can do it.
Yeah, okay, so I think what you're asking is, what I showed was trapped particles, but I said that we could figure out their diffusion, but we've trapped them, so how can we do that? Is that your question? Or, okay. So the way that we're trapping them is closed loop feedback. We're basically scanning a laser, and every time we see a photon, we know where the laser was, and so probably the particle's there, plus some background, right? And so we use a Kalman filter to estimate where the particle must be. And if it's this side, then we kick it back with the right voltages. Okay, and since we know the pattern of places where we saw the particle and voltages that we applied, you can back calculate how the particle is responding to the fields and how hard it's trying to escape the trap, which corresponds directly to its diffusion coefficient, basically. Yeah, so the simple version is if you trap a thing and it's really trying to get out of the trap, it's probably small. And that's, but you can be a little bit more quantitative about it than that. Yeah.
Oh, can I ask a follow-on question? So could you use that same methodology, if I'm understanding it correctly, to maybe assess? So let's say you want to pull. A lot of folks are fans of torturing nucleosomes in vitro, and so could you do springiness with that same concept, maybe? Yeah, because people are still trying to find direct evidence of, just how stable the different the dimers versus the tetramer, whatever, are in nucleosomes, or even readers, right? Reader proteins that come in and interact. Or maybe even a more general question is, have you applied your system to DNA protein complexes in vitro? Because, I really like that. You explained it really well. I think I kind of understood that, right? Where you could draw some conclusions about where that particle was based on how it is behaving after you trap it. I like that. Yeah, got me really excited.
Okay, short version, yes. Although I do think that optical and magnetic tweezers have cornered the market on force spectroscopy, so we don't really need to. We cannot do anywhere near as well as they do for all sorts of reasons. But we should talk because we're also thinking about how to poke things inside a cell with a very different version of the trap that I showed today. So, yeah.
So let me maybe poke on a little bit more. I was just curious. How stable do you think all these things that we write about in textbooks, we show pictures such as nucleosomes, DNA wrapped around nucleosomes, nucleosomal clutches, are they real structures or this is just statistical representations? Are we talking about, do we live, okay, let me rephrase it, do we live in the world of probabilities or do we live in the world of realities, real structures?
Yeah. I mean, I can only respond with the point that probably a lot of folks that are familiar with chromatin research in general kind of understand this point. Any data that we collect, unless it's like single molecule or single cell, is kind of like an average across a bunch of events. And I mean, I don't know. I mean, maybe the more mathy folks in the room could like dive into that a little bit deeper and determine how you can kind of use that imperfect bulk information to draw a conclusion about stability, frequency, because the way that like ChIP-seq data are presented in the literature, and I'm guilty, I participate too. Enrichment. Oh, there's more protein there. And it's like we're thinking of it in a totally wrong way. It's like this statistical representation of the frequency at which a protein had been sitting there when you did the assay. And so like a way to just kind of like really dig into that. And I don't know. I mean, so maybe that would take us in the right direction in terms of understanding where we're looking at things that are very stable versus things that are in flux. I mean, maybe just better ways to look at the data that we have now. I don't know. Any thoughts?
Maybe instead of asking how accurate our models are, we should be asking how useful our models are, and can we create actionable or like introduce new questions that are actionable based on our models, even if the models aren't fully correct. I'll tell you right now that the model I presented is not, it doesn't capture all the aspects of chromatin clustering. And if you're interested in what we leave out, I go into great detail in the discussion section of that paper. So there are a lot of factors we miss, but the factors we capture are useful enough to tell us how reader proteins play into this. So maybe just a glass half full perspective. Even if our models aren't fully accurate and they don't hold up over time, maybe they will have led to useful outcomes.
Well, this conversation kind of reminds me of a debate between Albert Einstein and Niels Bohr about cats. Is it possible that all this structural biology data that we grew up with, in fact, just realizes itself when we take the measurement? Right. All right. So now I understand. I'm fully cognizant of the fact that I'm the only one who is standing between you and drinks outside. So I want to be respectful of that. And with that, I would like to ask one more question. Let's play a game. Over, under. What is, in your opinion, the most important and impactful, most transformative, but underappreciated question, scientific question out there? Or conversely, what is the very popular, overappreciated question that's really not that important? And if you can combine both in the same sentence, over and under, I will personally get you an extra drink. Although they're actually all free, so it doesn't really, not going to cost me anything. But
I'll take the extra drink. I'll offer one. Okay. I mean, it's really specific, right? Because I've been looking at the same protein for a while. But so those CBX proteins, right, that I kind of talked about in my presentation, if you compare their in vitro affinity to like an antibody, their garbage is garbage. Any person that like measures antibodies in vitro, like nanomolar affinity, right? These things, micromolar, 5 if you're lucky, 500 or undeterminable. No, seriously, look at the literature. But so then I think that's the underappreciated observation. And then, wait a minute, how was this again? How did this work? Over, under. But the impact, I think, would help us to understand. Now, also, if you mutate one of these things in a mouse or a stem cell, the cell goes crazy. So how can a cell put so many important development and processes on these crap, weak, very transient interactions? And that's a huge question, right? And I think part of the barrier to really understanding is the context in which we study, right? We're looking at protein histone interactions and like PBS buffer or whatever we're using, right? It's not nucleoplasm, right? You're removing all the other proteins. So yeah, I don't know. So that's my own personal one.
I'm going to start some fights. And I'm going to start with the one that's going to start some fights, which is I have some hot takes on AI. I think neural networks, machine learning are fantastic for certain types of tasks. I love Fang's description of real-time figuring out the pupil function. That stuff is awesome. It scares me when I see talks that present AI on an image, because what you're actually getting is a computer drawing in for you something that isn't there but is kind of reasonable in the same way that a human might do that. So I think we have to be very, very careful in how we use that. Everybody probably knows that. Maybe that's not enough of a hot take. I hate ChatGPT, so that is a hot take. The thing that I think is underappreciated, I actually completely agree with you. I think that really weak molecular interactions and the messiness and crowdedness of the environment inside a cell is the thing that's so hard for us to grasp, but is so critical to the way that biology actually works. Weak interactions might mean rare but strong, or they might mean that things are bouncing off each other all the time, and that matters a lot. When you stick something inside a cell, a protein, whatever it is, other stuff gloms onto it immediately. This stuff is so hard to study, but I kind of wish that I had a better way to do it, because it's so important.
So I want to be very careful with how I answer this question because I'm on the postdoc market. So the way I'll approach this is what's something that's underappreciated outside of this room. And I think the role of chromatin organization in gene regulation generally is underappreciated outside of our community, perhaps. I presented work about my research in front of bioinformaticians or more data scientists who think there's more information to pull out of sequence data rather than histone modifications or ChIP-seq data. So I think gaining buy-in from those folks could help us to expand our focus a bit.
Well, this was fascinating, fantastic. Thank you. And I know one thing, that most likely when I go to bed tonight, I will not be able to sleep because I heard so many incredible ideas today that they're going to be kind of circling in my mind. Maybe I'll need to take a sleeping pill. I think the conclusion that I draw from this discussion is that world is a fascinating place. Genomics is a fascinating place. And especially young people in this room, you are lucky because you live you're probably going to work and have some discoveries that would be completely unprecedented. And it's an amazing world out there, unless AI is going to turn us into pets, right? And make us draw pictures for its entertainment. Right. And with that, let me thank again our volunteers, every one of you, for being an integral part of this event, making this a success, and of course our 6 incredible speakers. You made this day very special. Thank you.